Cool. So thanks everyone for coming, and thanks for coming online. If people are online and hopefully you can hear us, first time we've tried having this session be broadly available. So so welcome feedback. Please feel free to just chime in. We're in the room. There's like 15 of us here. So if if you want to ask a question or say anything, just feel free to speak up, and we can be happy to answer questions from everyone. I think we've been doing this group for 5 years, 6 years. I mean, it's more than that because I was at the age before, so it must be 6 years or something. It's sort of a local group, so if anyone's listening in there in the Boston area, you're always welcome to join this meeting. There's a list, and anyone remotely, we're trying to kind of open it up and maybe make it a little more broadly applicable to anyone who would want to see it. So with the caveat, this is kind of like a lab meeting, so it's mostly it's meant to be like, don't spend your entire life preparing for it, and it's a lot of preliminary results and just discussing it. So we'll really just talk about stuff we're working on and share and meet each other and stuff. So that's the broad overview. And cool. So I'll start. So I was going to talk about bcbio, which is what I always talk about every single year. I noticed from my past docs, which is cool. And for anyone online who doesn't know, I work in Boston, obviously the Harvard Chan School, which is we're at MIT right now, so it's over the river from here. And if you're interested in any of this, it's you can go. All these slides are available, and I'll put I'm recording it, so I'll put the recording up as well. But you can go to bcb.io, which has a blog post and other things described here, and then there's j.mp bcbio links as the actual slides and all that. And links from here. So what I'd like to talk through is start off with a spiel about open source communities and why I think they're valuable and why we work on that, and then specifically describe bcbio, sort of what it is, which is community developed analysis tools, and then talk through the value of hey guys, how's it going? Value of variant validation. So some examples of how I think validation is really useful, and that's how we do a lot at bcbio. And then talk about stuff we've been doing with interoperable infrastructure, which is trying to work together better using the common workflow language, and sort of tests and stuff we've been doing there. So the thing that motivates a lot of this work is in trying to support bioinformatics, right? So this is a BioStars, which if you've ever been there, is a real super useful community site for answering questions and asking questions. And if you ever go there, there's an unbelievable amount of questions and an unbelievable amount of work goes into answering those, right? And there's a lot of people doing a lot of cool things, and it's very hard to support them. And so it's sort of a challenge as a community. How can we work together to sort of train everyone up to help people that need help? And BioStars is sort of one answer. Another answer is communities and building communities. So one thing that I'm very involved in is the Open Bioinformatics Foundation, which holds a yearly conference called BOSC, which is not about pairs, which is about bioinformatics open source conference. So it's all about taking care of the community, working together, thinking of ways to work together. And as the open source community has been a big sort of thing for me over my career. I started with BOSC when I was in grad school, and it was really awesome. I was in a lab by myself, and it was cool to just be able to meet with people who thought like you do. And it's it's since BOSC been going for I don't know, 15 years, 17 years, something. And at the time it was kind of controversial to have open source stuff. And now it's some people are losing audio. Sorry, I'll blame the MIT networks. Hopefully it's coming back online. Let us know if it's terrible forever. And we get okay. Sorry, yeah, it might just be MIT gets mad. Well, if it's really terrible, I can try to switch to a different network. I think we're on the MIT guest wireless, so. But it's all good. But yeah, so that's open source community is awesome. And so a lot of things I'm interested in is sort of how you can build communities and work together. And this is just a slide to sort of you know, just have the word community really big, but it's also an awesome book by Peter Block about how to sort of build communities and bring things together. And so the reason to build communities, I think that working together produces awesome things. So this is the about page of participants from the Nomad project, which I think is how you pronounce that, which is formerly the XAC project. And the point of the slide is that there's so many people on this slide you can't read it, right? So there's this awesome group of people that work together, you know, both PIs, projects, you know, production people to build this awesome resource called Nomad, which is, if you haven't used it, is a great resource to help figure out if the variant that you found is in any population anywhere, because it's a collection of tons of populations all processed and analyzed in the same way. And this happened because of putting together a community of people who care about doing it and building infrastructure around that and supporting it. And so that's a really cool thing. And the challenge, I think, is that there are many communities. So within, this is just an example of 3 sort of really good communities within the open source. You know, Galaxy and CBio is an awesome portal for cancer genomics, and Synapse is an online collaborative repository as well. And so there's all these different communities, and what ends up sort of happening is that the communities get broken apart, right? So if you're working on Galaxy and you're doing things with Galaxy, you're part of that awesome community and you're working on things together. And if you want to start, but then if someone else is working on Synapse and doing their analysis there, they're part of a different community, right? And so it's very hard for those communities to end up talking. And so bioinformatics and especially open source, people who want to do this stuff, is a small community, right? So it'd be awesome if we could all work together. And so I'll come back to this slide later, but you know, sort of my feeling is that open source communities really aren't yet optimal. So this is a fitness plot that you can, you know, pretend like the, you know, higher axes are, you know, more better science happening, and lower axes are, you know, less efficient science happening. And you know, for a lot of these projects, we're at sort of these local maxima where you know, you're I don't know. So I guess it's starting, you know, you can start at the bottom, way down at the bottom, and you know, you're at like this low point where you don't know what you're doing, and you're working by yourself, and you have no friends to help you, and you're just trying to struggle through. You know, and then you join a community, and you can become more efficient, and you know, people are working together, and you reach these sort of local maxima where you can do a great job. But then what happens is you're sort of stuck in this, right? So I'm in this maxima over here, and I'm working with Galaxy, and I want to like do something that's over, you know, that the Synapse community is doing over here, and it's really hard to do that because in order to like connect those two things, there's this big loss of productivity trying to connect them and get things working. And so you end up sort of within your community. Ideally, we'd be up at some, you know, assuming there was a global maxima, right, of productivity in bioinformatics where everyone's getting along and super happy. So the question is sort of how to get there. And I think like one place to do that that's pretty awesome is in large scale infrastructure development, which is stuff we work on. The reason I think it's a good thing to work on is that it's a shared problem. So it's, you know, it's not specific to any particular research question. It's, you know, it's academics, industry, startups all have the same sort of issues around like building and running things. And so what that helps build, and we try to do in bcbio, is build these community developed analyses that give us the validation and scaling and ultimately support a big community of users who can work with the tool. And the way I describe bcbio sort of, and this is my picture slide, right, is as a white box software. So black box software is something where, you know, you plug in an input, you get out an output. And I think that has a ton of value, right? Because, you know, you don't always need to know all the internals that are happening inside. Like sometimes it's nice to be able to call variants without having to like know every tool and put them all together. And that's a really good like level of abstraction. But you also sometimes do need to dig in and know what the internals are. And, you know, as a scientist, you want to know what's happening. And so you want to be able to look inside. And so this is, you know, this is a picture of a Raspberry Pi inside a clear box, right? And so that's sort of meant to be provocative of what bcbio is supposed to be, right? You can, you know, you can treat it at this high level and just plug things into two sides and things happen. And but if you want to dig in, you can open up the box and see all the messy, I mean, beautiful internals and see what's happening. So So I guess to make the concrete, what bcbio does, if you haven't really used it before, is it takes like a set of sequencing samples and a configuration file, and it integrates a whole bunch of tools. It scales, tries to be resilient to failures. You could restart, use all the kind of stuff you'd expect from like something running a pipeline to do. And then on the other side, you get out, if you're doing variant calling variations, so SNPs, indel, structural variants. We have RNA-seq, we have small RNA analysis, so I'll talk mostly about variation today. I think Lorraine was talking later about small RNA stuff, so I won't talk about her great work. And Rory does a ton of stuff on RNA-seq in our group. So we have all that stuff in bcbio, a lot of pipelines. I do most of the variation stuff, so I'll talk about that. And then we also try to tie in with other tools, work together. So, build quality metrics, so you know how your things did. Feed downstream analyses, so you can annotate and query and visualize things, work together. And so if you, I feel like you have to have some code, right? So this is my YAML page of code. So this is what you put into bcbio, and this is sort of the high level configuration. And so you can see like it's really at sort of you're thinking at a high level, right? Like, so you want to do a variant analysis. This is variant 2, because variant 1 was a long time ago and is a terrible pipeline. So we have variant 2, which is now the same as variant 1, because variant no longer exists. But it's a variant analysis, right? You're doing doing an HT38 genome build, and then you're just really defining at a high level. Like, I want to align with BWA. I want to mark duplicates. I don't want to recalibrate and realign. I want to call with free variant callers, haplotech caller, free base and pardict. I'll do an ensemble on those. If 2 or 3 pass, I'll keep them, and I want to do 2 structural variant callers, lumpy and manta. So super high level, right? You're just saying like what you want to do, and then you feed that to bcbio, and it happens, and you get your outputs. That's the idea. And so just to make concrete, like sort of the type of tools we use. So bcbio doesn't build aligners. We use aligners, right? So we're using BWA, NovoAlign, Bowtie, HiSat. We're, using variant, variant callers like FreeBase and GHK. We're using RNA-seq tools, the quality control tools, and all the manipulation tools. So we're really trying to put together a whole collection of tools and make them work together, not build stuff that already exists in that way. And so practically what we do provide is a community, so a collective set of expertise and installation of all the tools and data and other things, so that everything works together. That sort of was the history of bcbio. Was like, that was sort of the initial thing that we did that was different. Is I built this, I was working on Cloud Bio Linux, which we still use for installing this, and we made everything install automatically, and that was pretty cool. And then people could actually run the pipeline because they could install it, and so that was sort of step 1. And then it's evolved over like 18 different steps since then, and now we use this Bioconda project, which is awesome. It's a collection of people building tools, so that's a really useful thing outside of bcbio as well. Sure, we'll link to that in there, but it's Bioconda, and it's a great group of folks. There's a ton of recipes. It's an installer. There's almost every biological tool you can think of is there. There's a big community that builds them and installs them. They're pre-builds. Works on Macs and Linux, and it's pretty nice. It's made the installation tons easier. And so we do that. We integrate all these tools together so they actually work, and then do validation. So make sure that things work. And I'll talk more about. I think that's sort of the critical thing or cool thing that we do in bcbio. So I'll talk more about like how that looks for a germline evaluation, and then scale things so you can run on big samples. And so. Yeah, so that's a bunch of talk, right? And then you made a pipeline. So what, right? This is a depressing paragraph from a CPipe paper, which is a great tool for clinical informatics from the University of Melbourne, and it lists sort of all the different sort of other tools that people have. So it's, again, you're not really supposed to be able to read it, right? There's a lot of tools that do it, and sort of why they're not quite right for what they needed, and so they built their own tool. And this is sort of thing that hopefully we're trying to avoid, is by putting something that can all work together. And so the thing I think that's important about pipelines that's different about bcbio that we try to make different is that it's sustainable. So this is a definition of sustainability from Software Carpentry, which are an awesome set of folks that do training materials, and we do software carpentry. I think you guys do them at MIT too, right? Have you done it, Charlie? No, our software carpentry. But well, we do them, I guess. But we have software carpentry come in once in a while and do training here through Harvard Chan School, and our teaching team does an awesome job with that. And they're just a great group of people trying to help with like training. And this is a good quote, I think, that's not relevant to training, but really relevant to how to build a tool, right? Is that a piece of software is being sustained if people are using it, fixing it, and improving it rather than replacing it. So the challenge, really, especially in academic science, right, is to keep a tool going for a long period of time. So, you know, the reason that's so challenging is that things change all the time. This is my GATK changing slide. So it dates how long I've been doing it. I have a slide that the next slide dates it, I think, how I've been doing it. But, you know, when I first started some iteration of this, it was just like, oh, couldn't you get this GATK thing working? There's this, you know, and they had a pipeline that was version 1, and you know, so we got it working, and then that was like, oh, that's gone. Now it's version 2, and that was gone. Now there's version 3, and then there's version 4, and then version 4 was gone, and now there's a new GATK 3, and you know, soon this will be updated because there's GATK 4 is coming, which will be much different as well. And so that's awesome, right? It's awesome. Every 6 months, you know, the GATK team is reinventing and doing the things and moving things forward, which is great. It just means that if you're doing this yourself, like every person, every pipeline has to keep up to date, right, and update all these things, and you know, make sure the new version works and validate that it works. And it's a ton of work, right? And it's hard to scale. And so what we've been trying to do is keep bcbio going for a long time. So this is the depressingly long slide about how long I could give talks about bcbio, right, for 6 years. Been trying to do this and sort of pretty consistently. This is a graph of contributions on GitHub. So, you know, if you have green and things are up, then you're working. If you don't have green, then things are, you're not working. And so we've been pretty consistently trying to maintain it and support it over time. It's not just myself, it's an awesome group of people, both in the core. I mentioned Lorena and Rory do a ton of work. And then we have a great group of community of people that work on it as well. People are working on it, which is awesome, and we're keeping it going. And part of that is support. So there's 1,250 issues. I think there's more now. I'm sure we're at like 1,600. So those are all just questions, right? There's never any problems with bcbio. It's only just questions. But no, I mean, so there's issues, there's things that you fix, there's questions, there's all kinds of things. And you know, it's a challenge sort of how to support group people, keep it working. And that kind of thing. And that's one thing we really try to do a good job of is, you know, even if you can't fix or know someone's answer to someone's question, at least you can try to help them get to there. And so we have support. This is on GitHub, and we welcome contributions. We've had lots of people contributing both fixes and code and everything. So, you know, please, if you're interested in this kind of thing, go there and check it out. And everything is documented and you can get it going. So, you know, we have getting started documentation, how to install it, automated installer, you know, example pipelines, all kinds of things. So hopefully you can get it running, because if you don't know if there's no documentation, it's very hard to get going. And so, you know, we support, I'm going to talk about variant calling today and sort of the infrastructure stuff, but we also do RNA-seq, single cell, ChIP-seq as well, small RNA seq. So lots of different things in there. It's cool for playing around, and hopefully we're always talking one time. You can talk about the RNA-seq part as well during this. Would be sweet. So that's sort of bcbio and trying to sell it, right, for 15 minutes, right? So now we can talk about science-y stuff. So I think the first thing that I think is the coolest part that we've done in bcbio is spend a lot of time working on validation. I started doing this, like, I was given talks about this, about the X Prize project, so people who have been around for a while probably remember. And that was sort of the initial impetus to sort of develop, you know, validation frameworks for variant calling. And that's evolved into a bunch of different cool communities that work on that. And it's really why I like working on burn calling, because there's actual true sets, which are imperfect, right, but a way of describing how things do, and then you can actually test your pipelines against them and know how they do. So having validation lets you, essentially, I think of it like integration tests for pipelines. So if there's a new, if I change things to bcbio, where there's a new release of a software tool, is it better or worse than the previous version? It's very hard to know that, right, without just being like, well, you know, the GHK team is always right, they have a new version, it must be better, and just putting it in. So what validation lets you do is actually test that assumption, you know, trust and verify, right? So, you know, you can put it in and validate it and make sure things actually work. It also lets us do unbiased algorithm comparisons. So, I mean, I don't really care which tool works the best as long as I have one that's working the best. You know, so it lets us sort of be a way to describe and compare tools to each other, and without, you know, having to make one better than another, just try to understand what's best. And it's also a baseline for improving methods. So you can sort of understand which part of the methods are useful or not useful. You know, one thing that we did a lot with was with recalibration, realignment. I know GHK team has backed off a lot on sort of their recommendations for that. But it used to be like, you have to do recalibration, you have to do realignment. And, you know, we did a bunch of validation. We were like, well, you know, it helps a little bit, but this is how much it helps. And that gives you a sense of like, if you can plug things in or out of your pipeline and what, you know, what's very useful and what's not. And I think what that lets you do is make changes and fix things. Otherwise, you're sort of stuck, right? You're at some version that worked and someone said it worked, or it worked for some particular project in some way, right? And then now you want to change it. Is it going to be broken for that old project? I don't know, right? And so this gives you a way to get out of that mire of like, I have to use the exact same version I've always used because it worked one time, and actually move forward. And so the value, I think, of this is become building these reference materials. And so the Genome in a Bottle consortium is sort of the thing that developed out of the X Prize, which is an awesome group of people working on building reference standards for variant calling. And so any 12878 was sort of the initial, and that one in a second. So the initial test set, it's like with sequence, everyone sequences a ton of times, is testing their sequencers, and so there's been a ton of data about it. And sort of they put it together and made a true set, and they've been iterating on that true set ever since. And this is kind of rolled into the GA4GH, the Global Alliance for Genomics and Health, which is a community of people who just want to sort of help work on making things talk to each other. And that's been an awesome community that's sort of taken over some of the benchmarking work, and sort of Genome in a Bottle and GA4GH work together pretty closely. Well, Justin Zook's in charge of both groups, so he makes them work together pretty closely. And then there's been on cancer side work as well, like the ICGC TCGA Dream Challenge is cancer data sets, and they've had, I think they're on their 7th iteration of different challenges and true sets that they've been working through. So that sort of, if you, without those reference materials, you can't do validation. So it's really super important to have those. And so I'll describe how we use Genome in a Bottle here with bcbio to look at GHK. So this is a collaboration we did with the GHK methods development group. They were interested in having someone else benchmark GHK haplotype color. So we did that with 3.6 and compared it to some other methods. And this is a germline validation. And we did with the Genome Bottle reference materials. And I mentioned the NE12878, that's sort of a most famous HapMap sample. You know, it's a Caucasian female in this, you know, from Utah with 12 children or 14 children or something. That's been a really, really popular one to validate and benchmark on. But sort of over time, people know about that one and they're able to do a good job on it. And so it's good to sort of have a broad diversity and not over specialized to you know, people from Utah, Mormons from Utah. And so Genome in a Bottle has been introducing sort of more of a diverse set of reference standards using data from the PGP. So the Personal Genome Project has a lot of like freely consented open genomes that they can use. And so they have detailed true sets for 2 other ones now. There's the Ashkenazi Jewish trio and a Chinese trio. So you're starting to get where you have multiple data sets you can test on. if your methods work well on multiple different data sets, then you can be more confident that they'll work broadly on a broad end sort of data set. And so I'll show a few graphs, and this is my example of what the graphs look like to kind of describe them. So what I try to plot is sensitivity and precision. The sensitivity is on the left side. Sensitivity is over here, right? And precision is over here. And it's hard to plot sensitivity and precision because hopefully they're high, right? And so if you're at 98% and 99%, and you plot those they look the same, right? So you get this useless graph where things look almost flat at the top and are mostly filled. So to resolve that and keep plotting from 0, so instead of truncating the graph, what you do is plot 1 minus sensitivity and 1 minus precision. So that's the false negative rate and the false discovery rate. So if the plots are cool, they look nice. The only sort of caveat is that lower values are better. So this is it's just made up here. So method 3 is better than method 2, which is better than method 1 in terms of sensitivity. Got no audio.
I do.
What's that?
I do.
Oh, really? Okay. Oh, you're like checking in.
Yeah.
This is awesome. Marina's like next to me. Checking in.
Weird.
Okay. Too weird for you. Okay. But yeah, so yeah, lower values are better. So method 3 is better than method 2, which is better than method 1. And so that's kind of how they look. Sensitivity, precision, and they split into SNPs on the top and indels on the bottom. So SNPs and indels probably have very different properties in terms of their states. So it's kind of have those separately. So yeah, I have a couple graphs like that. So it's just to explain what they look like. And here's a real one that's not made up data. This is any which way to 8. This is the most popular validation set. And here's haplotype caller here, GHK haplotype, and you can see it does really well. This is SNPs and indels. It's on SNPs, it's about as good as SAM tools and FreeBayes in terms of sensitivity, and then in terms of precision, it's doing better, a little bit better, about twice as good as SAM tools, and a little bit better here than FreeBayes. And then this is indels, and so again, haplotype callers are really good. FreeBayes is doing well, and you can start to sort of, and the useful thing here is, right, you can compare these and see how they do in this data set, and you can start to see, like, okay, platypus is doing well. I mean, this is sort of, everything is relative, right? So platypus is doing a great job, just not quite as good as, haplotype caller, and and you can see, like, SAM tools here has some kind of false positive issue apparently with indels. So these are things you can pull out of these graphs immediately and say, sort of, if you're using these, this is a caveat, right? If you find indels with SAM tools, you might be more concerned about false positives. And if something, if you wanted to spend a month, you could dig in and figure out what's going on and why they're false positives and submit a bug report. That would be a useful community thing to do that I didn't do yet, instead of just talking about it. The cool thing is now you have multiple data sets, so you can run the same thing. So this is, any 24385, which is the Ashkenazi Jewish trio. And you can see the results look pretty similar, right? So, here's haplotype caller and FreeBayes, and, they're doing really well. And SNPs and indels, platypus, again, is not quite as good as SNPs, and you can see the SAM tools, false positives. And so you sort of have this confidence then that, okay, it works on these multiple data sets. It's not anything specific about 12878. You have a pretty useful comparison. And so, that's, I think validation is awesome. And so, I think what you're sort of seeing here is sort of just one practical example of how that works. So, you can see haplotype caller does well. This was some are doing with the Broad folks, so they're happy that haplotype caller as well. I think most people know it does that. FreeBayes is also a very good caller if you're, if you have licensing concerns with HK. That's another really good caller. They're consistent. All the callers are consistent across diverse samples, which is awesome. And then we identified some potential areas you could start tuning and filtering, improving filters, right? Like SAM tools and platypus. And if you're interested in, like, sort of that tuning, we have stuff on the blog that I mentioned earlier, bcbio, where we try to tune it where we have tuned bcbio for different all the amount of work that goes into that. And, you know, there's been a lot of cool competitions recently that sort of help do this similar sort of thing, which is really nice. Like the Precision FDA is a recent challenge where they put together these true sets are ones they used
blue collar bioinformatics. Yeah, okay. Mm-hmm.
Platforms, and it was pretty sweet that they could you know, you could contribute and really see how things are doing and sort of build up those you know, a community of people doing this sort of validation, not just within bcbio. So, you know, not just pretending it's me doing it, it's an awesome community of people working on all these things, which is sweet. And so I wanted to kind of switch that sort of bcbio validation. I want to switch to talk like technical stuff about infrastructure and what we've been trying to do with infrastructure. So I started by like listing what we'd like to do with infrastructure in bcbio. So you want it to be free, open source, community developed. You want everyone to be able to work on it. You want people to be able to contribute to it and help them contribute. You'd like it to be able to run sort of everywhere on local machines, on clusters like Swarm, SGH, or PBS, all that. Since we can't just have it one scheduler, there's got to be 10 different schedulers you have to support, right? You like it to run in different clouds, Amazon, Google, Azure, and it should run in clinical environments. You should be able to have this nice user interface that researchers can use. It should integrate with LIMS systems, so you can automatically come out of there. You know, the general public should be able to take this tool and run it. You know what? And so that's you know, we're never going to be able to do all that stuff, right? Like that's all sounds awesome. It's not doable by any one set of people, right? It's a ton of things to do. And so this sort of goes back to this graph I showed earlier, is that, you know, we suffer from the same problem any other community does, right? And that once you start to build up infrastructure and things around a tool, you're sort of get stuck within that local maxima. And so, you know, bcbio has its infrastructure. It can distribute on a cluster. It works well. But if you want to interoperate with something else, then you have to build the interoperation to do that. If I want to run an AWS, I have to build the stuff to do that. And that's not the way we want to do things, right? Obviously, we'd like to be able to work together with people. And so what we've been trying to do over the past year really is try to build better abstractions that would let bcbio interoperate with more tools and do more things. And so the sort of, I think, two biggest ones have been the common workflow language and Docker. And so I won't talk more about Docker, but if you don't know Docker, it's like a containerization framework. So you can stick all your data, all your tools into this container, which is just like really just a fake sectioned off section of your file system, right? But you can take that and ship it around. So, you know, bcbio will install, and it does a pretty good job of installing, I think. But, you know, this makes it way easier. If you want to bootstrap on a machine in the cloud, you can just download this Docker container and run it immediately. And so what it's done is push the isolation, so all your tools are working a certain way in a certain place. So that's been super useful. And then once we had that in place, the next really issue was that the way we describe things run on a cluster is a barrier to things working together. So like bcbio has its own sort of not even really a workflow engine, right? It's like a set, it's like a descriptions of parallelization that can happen. It's, you know, and we use IPython under the covers to actually do the running. And so, you know, we're building on top of things, but, you know, our way of describing things is not compatible with anyone else's way of describing things. Just like most people's way of describing things is not compatible with anyone else's way of describing things. And so this project happened 2 years, it started 2 years ago at the at CodeFest in Boston. We have this, I mentioned the Bioinformatics Open Source Conference. We also have this 2-day hackathon, CodeFest working session beforehand, where we get together and just work for 2 days on open source things. And a group of people came together and started working, saying, you know, it is kind of silly. We have all these workflows and we don't have a way to talk between them. Can we build something that does that? And so the common workflow language is what came of that. And it's been really cool to see, because it sort of has built out of that community. And this year at CodeFest, people were there from the common workflow language, and they had a version 1.0 release. And, you know, it was pretty cool to see sort of the community coming together and building this. And I mean, practically what they do is, this is another, I should have started with this one, so I'll start with this one, I'll go back. Sorry. But what the common workflow language does is it describes workflows. So essentially, you just have these it's a technical specification of a language, right, that describes like how you chain steps together. So you have a workflow, and you can have sub workflows, and you have steps within those. And each of the steps calls out to some kind of tool, and you have to describe how the command line calls out to that tool. And so there's a whole set of specifications around how you can do that. And the cool thing and what happened is that every single project that does workflows had their own sort of way of doing this description themselves, right? Like, so they all had their own tool definition language, they all had their own workflow language in some format or another, and none of them could talk to each other. And now you have this like single target that you can work towards. And there's, of course, there's not only one workflow description language, right? Because it's science, right? Everyone has to have their own workflow description language. So there's others as well. The Broad has been working on a similar problem, and they have this language called WDL, which also describes workflows and tasks, and it describes how to parallelize things, just like CWL does. And they have slightly different focuses. So CWL has been a little around sort of the interoperability, working in multiple places. WDL has been around sort of having a user-friendly language that you could write. It's sort of born out of it's from the Broad, and they are trying to support a community of users there who want to write their own tools, and so they wrote a language to do that. And so these are sort of projects that help. What I think they do is help describe important abstractions. So there's a question of like what's better, WDL or CWL or any other thing you can make up, and really they're the same. They're equivalent in almost every way, right? Like they just describe things, tasks that happen. They have different ways of presenting that to a user, but in general, they cover the same ground. And but I think what's important is not like which one is better. It doesn't really matter in the end, right? And no one's ever going to agree, right? That's like trying to agree on like a presidential candidate or degree, anything. You get 3 developers in a room and talk about something, you're going to get 5 different options for how to do it, right? So, the important thing is that we're working at the same level of abstraction, and I think that's much more important than the implementation. And so, what we're trying to work on in bcbio and sort of in general is defining this abstraction. This is a layer at which we abstract our tools and work, and then we can make things that run in all those different places. And so what we have in bcbio to do this is right now we have CWL support in bcbio. So if you used bcbio before, it's essentially starting off is the exact same as you do for running a tool. You describe what you're going to run in that high-level configuration file I showed earlier, exact same file. But now instead of saying like bcbio run this file, what you do is say bcbio generate CWL, and it generates a set of files, CWL files. And then you can take those files and run them on sort of any infrastructure that can support CWL. So all you need to do that is the CWL, some way of like running the tools, and that can be either Docker or a local bcbio installed on your system, and then the genome data that links to. So what this is doing is letting us run anywhere, right? So the reason it's a pain to do this, right? Like, I have to rip out all the internals of bcbio and convert to a different language, and it's hard, right? And so why do that? And the reason is, and I'll talk about all the different slides, that you can integrate with multiple platforms now. So now we can run on our Vados, we can run on Toil, we can run on Cromwell, Galaxy, Nextflow, 7bridges, DNA Nexus, all these different places, because we built this abstraction. Like, without building an abstraction, it would be impossible. Like, you would just go to someone and say like, oh, can I run on this? It'd be like, no, there's no way, right? You're not going to like re-engineer all of bcbio to run on this specific thing. And so now you have this infrastructure that you can do that with. So I think that's been really awesome and a way to sort of move bcbio into integrating with these places. And the goal is really to stop maintaining the bcbio specific infrastructure. So hopefully we'll no longer have a way to run things in bcbio. It will just generate this tool, and then you will pick your favorite platform to run on, depending on where you want to run it, what you have on your system, like where you're running, and what works best for you, and you can do that. And then the bcbio community works, and they work on building up a better workflow system and that kind of thing, and bcbio works on solving the biological problems underneath to run things. So that's the idea. So I'll talk through like sort of the practice. So as we have this, it works in practice. You know, you can go to that page and you can see it, and there's test examples and all that stuff. And so some of the places it works in is, the first place it ended up working was in Arvados, which is a cool group of people. They're in Davis Square. They're a local startup in Davis Square, and they build this open source tool called Arvados. They're called Pureverse is the name of the company. We worked with them and they're sort of one of the initial founders of CWL and have contributed a ton to the specification and implementations. This is sort of the first place we got bcbio running, working with them, and it was with CWL, and it's pretty cool. You can go to, they have an open site called cloud.pureverse.com that you can get an account on, and you can see the bcbio runs there, that I often run there and do tests on that system. So it's pretty cool, and it's you know, it's amazing. It's amazing to have it be able to work in some like distributed environment like this, whereas it never was initially designed for that. So that's been really cool. It also works with a workflow engine called Toil. This is from the UCSC Big Data. They have a long name. It's the NIH Big Data to Knowledge Center for Translational Genomics, BDK, I think they call it for short. But they have this workflow system called Toil, and that supports CWL. It essentially takes the CWL, it ingests the CWL as you produce it, and it converts it into their internal Python workflow description and then runs it. And one of the reasons we've been working on Toil is because they have local HPC support, so you can start running on Slurm and SGE. I mean, Arvados is mostly a cloud platform. That's their primary target right now. So it would be hard to, you couldn't just automate the installation of it on a local machine like we do right now with bcbio. Whereas this might be something we could use for that. And also, Toil also works on the cloud, so they have a ton of work with AWS and Spot instance. I think it's sort of where they put in the most time. I think it also works on Google and Azure. But so already this is pretty cool, right? Like it works in 2 places, like that's, you know, and then sort of the other things we've been working on are sort of more in progress, but you know, I think they're happening. So the one thing we've done a bunch with is trying to sort of bridge the gap between CWL and WDL. So Broad has this tool called Cromwell, which is their engine that runs WDL, and it's nice. It runs on Google Cloud. It runs locally as well, and so it'd be, you know, cool to run on that, and also try to demonstrate how to go back and forth between CWL and WDL. And so we've been working on trying to take the bcbio workflow abstractions and convert them into WDL so they can run there. So both WDL and CWL have sort of their own things that are hard to convert, but there's sort of a base set of functionality around just like describing tasks and workflows and nested workflows and parallelizing and grouping and batching samples that interoperate pretty well between the two. So that's sort of where we've been trying to focus. And I think hopefully by demonstrating it, get the communities to talk more about sort of both what they need for their particular workflow language, and then what's a interoperable set. So if you want to write your own interoperable set, you can do that too. It's not just bcbio. So that's the Cromwell, WDL, Broad. We've also been, you know, the Galaxy team has been working on CWL support. They have, if you use Galaxy, they have tool descriptions that are XML files, but they're functionally equivalent to CWL's description of tools. They have a workflow description language, which I think is also an XML. That is also functionally equivalent to CWL. So there's a lot of work, right? Like they've built a lot of things around this infrastructure, and so they've been trying to work on converting to run CWL. And I think right now they support tool definitions for CWL, at least with this Planemo tool. That's a runner for Galaxy, and they're working on workflow support as well. So I'm a huge fan of Galaxy, and I talked about that a bunch of times at the big meeting too. So this would be awesome. This is like my dream right here, is to finally be able to fit bcbio back into Galaxy. So that is hopefully happening sometime soon. We also just had a call. This is from Twitter, like last week. That the Nextflow, which is another workflow engine that's pretty nice. that they were thinking of working on a converter between Common Workflow Language and Nextflow's source code. They have sort of a domain-specific language like Cromwell and Whittle do. So I called with them this week because I thought it was awesome, and I said, like, can we all do stuff with it? And so hopefully, that would be the same sort of conversion as what's happening with Whittle, is you describe some subset of things that are allowed to convert between the two, and then you can have a converter that makes those conversions. And so this is cool. It's good to have that's another thing that's sort of upcoming. Seven Bridges, they're also they're a commercial company. They're here in Cambridge, and they're kind of all over. They're in Belgrade too as well. And I know they have European stuff as well, but they were one of the initial people that worked a lot on the CWL specification, and their internal workflow description uses CWL. They sort of had, when they were having to do actual real work with CWL wasn't a place where you could do actual real work. So it was still like sort of becoming a specification that you can work on. And so there's a version 2, or I guess a draft 2, I should have said, instead of version 2, because that sounds bigger than 1.0. But they had draft 2, that's workflow draft 2, plus these extensions that they sort of need to actually get work done. And so I mentioned as a summer at the Code Fest, they finished version 1.0, which is sort of a finished, at least a target version that can work in real life scenarios, does like all the things you need, and like sort of supports the extensions Seven Bridges have. So they're moving to that. So hopefully, as soon as that is moved, what bcbio has will run on there. And they were just at a hackathon yesterday in London, and supposedly they have a runner. It's called Bunny. They have this whole like rabbit theme going on with their tool names. But they have a runner that supports 1.0 and supposedly can run bcbio now. So I think we're nearly there. So that's awesome. So you're talking about another place where you can run it as well. And then we've worked with DNA Nexus as well, thinking about how to sort of fit this in with their internal workflow language. So right now they can integrate bcbio by its Docker containers, like it can sort of suck in the Docker, and I think they have their own sort of I don't totally understand all the internals, but I think they have their own like image format that they're using internally that it just converts from Docker into those. And then to run, you can run bcbio on single nodes, and scale things up that way, which is a pretty nice way of doing it. But ideally, over like a long term, you'd be able to sort of parallelize things like you do in bcbio. And so the idea that there's some initial code to do is to convert CWL into this DNA Nexus API. So you distribute it that way. So it's kind of similar to what we were doing with Whittle or with Nextflow, is you're actually taking CWL as a target language and then converting it into a domain-specific workflow language. So I think that's all. The section. That's a lot of things, right? I think it's pretty cool, right? Like, so this is my wrap up. This is a pretty cool slide. Is that you're starting to build these connections between isolated things. So it's like it's still amazing to me that you can take it and run it in these different places. And so I'll probably not be bored with that for a while, seeing it run in a random place. And I think the key to that has really been like, here's an abstraction we can work with, and then okay, now we can actually do things. So that's been really cool. And so building these connections has sort of been what we've done a lot of work on in the past sort of year. And so since I talked about bcbio last time, right? So there is a summary, and no one asked me any questions. I've just been talking for like an hour straight.
Actually, I have a few.
Yes. Oh, geez, why did you yell at me? Yeah.
Yeah, I'm a new bcbio user,
Yeah.
and I really like the tool. I think it's wonderful. And just now you talked about Whittle and the Docker, and I think it's a cool idea to how to integrate bcbio into a cloud and a new system. I'm wondering, how does it handle situations, for example, GPU does not allow a free distribution? How does the Docker Whittle handle those type of situations? Yeah,
for Docker, there's a separate sort of command that we have in bcbio that will like ingest the jar file and put it in the right place. So you download the Docker, and then you have a second step where you second step where you add it into the Docker container yourself because we're not supposed to redistribute it. So there's an extra step, I guess, is the short answer. And then it just remakes. So Docker is just like a set of, you can just re-snapshot the container after you add it in, then you have it yourself, and then it's good to go.
Okay, I see. And also, you mentioned about the validation data sets when you evaluate the journal communications. Can you give us some recommendation about cancer validation data sets?
Yeah.
To evaluate the tools.
Yeah. So there's the ICGC is TCGA has a stream challenge.
Maybe they want to repeat it.
Oh. Oh, sorry.
Maybe people don't.
Yeah, I don't know if it's hard to hear the question. Sorry, we have a Jabra thing, but the question was around like sort of validations for cancer, and yeah, we've been using the ICGC TCGA Dream Challenge. They're synthetic data sets, but they're pretty useful for
It's in Dream.
Dream, yeah, Dream, EAM. That was one of the links. On the validation page, I have a link to where that is.
Right, so it's a simulated data sets, and we know the answer where is the mutation of frequency of abundance, and we can just compare the results.
Exactly. Yeah.
Yeah. And my last question is that I read in the manual of bcbio, and in the drawing the color section, there is a sentence. It says only use drawing the color if you have more than 100 samples. So why? What if they don't?
Sorry, use which one?
If you have only use join the color, haplotype, join the color.
Oh, join calling. Oh.
If you have over 100 samples.
Yeah. So if you have less samples, you can just call them. I mean, you can just call them all together in one pool. So you don't have to like do joint calling.
So why is it helpful if they have over 100 samples? Is it just the memory?
Yeah, it starts to get memory constrained. Yeah. I mean, so joint calling is useful, I guess, in two cases. One is if you want to like add in more samples later. Then the GBCF, so GHK uses an intermediate format, GBCF, to like represent the variance. And so you can, if you wanted to run 100 samples and you're going to have like another 100 later, and you want to call them all together, you could use those intermediates to call them without sort of having to start over from the beginning. You know, if beyond that, yeah, it's mostly just like a scaling issue. Like if you can't, it can be hard to run like a ton of samples together. So that's the only issue. And for GHK, if using that, mostly they recommend using joint calling. Some of the other callers, most of the callers, joint calling isn't as well figured out, right? So there isn't as much work on joint calling as there is in GHK. So for those, it's better to try to call them together as much as you can because the GBCF formats are sort of in progress when it's being done.
Okay, so and you have compared the results. If you have less than 100 samples, whether using join the color or not, join the color, the output is very comparable.
Yeah, we have a validation. I mean, it's not on. It's like on it's on the any 12878 family, so that's like 15 individual or something. But yeah, they look similar in terms of their outputs. So yeah, so it's not really a quality thing. It's more of like a making the analysis actually work sort of thing, you know. So yeah.
Okay, the very last questions that I see there's a contrast option in the configuration file. So if you want to filter out a bunch of variations, like you use contrast and provide the VCF file, including variations to filter out. But I don't see further description. Can I include multi-samples, multi-VCF files in the contrast option?
Contrast.
But I think that the background this year.
Background, yeah, background. That means
the
variations.
Some variants for the.
Yeah. I don't think that actually works. If that
Yeah, I don't know.
Yeah.
I don't know. I
Yeah.
never tried that one.
I was like, I don't know if that actually works. That's super cool. Yeah, no. Yeah, we don't really have a way to do that right now. There are parts where you'll where you can use sort of where it uses a reference database, so it'll use like exac I mentioned to like if you do tumor only calling, it'll try to use exac to find things that it thinks are germline because they're in those public databases. There's things like that, but not sort of a one-to-one comparison. That's sort of like it's it's a hard balance to figure out like what makes sense to be in bcbio in general and what makes sense to be like a downstream processing, and that's probably more of a downstream processing because it is like it'd be hard to generalize, I guess. You know? Yeah. Cool. Sorry, you guys have to save up all your questions. Yeah.
So when you talk about validation, what is your process standard?
Yeah. So we've been using these the genome in a bottle samples.
But those are already called. So
Yeah.
It's already known the variants then?
Well, I mean, known in the sense that there's been a lot of work on trying to put that together from multiple data sources. So like genome in a bottle has, like they collect, people sequence it from on a bunch of different platforms, and then they have a consolidation method that tries to put all those calls from all the different platforms together into one set of calls. So it tries to be unbiased in terms of like sequencing platform and caller and all that. I mean, it's imperfect, right? It's still like, you have to be able to know how to call it, and you know, different technologies we have, but I think they do a pretty good job of being general in terms of they're trying to avoid being like Illumina specific or GTK specific or anything like that.
So then you use a subset of that data, or
Yeah.
or you use a subset of the data but things that they haven't used?
Yeah, so I mean, we're just essentially like taking that as like sort of the baseline, right? So that's like the set of, that's supposed to be what the actual calls are, right? I mean, with the caveat that it's not perfect, right? But you have to sort of treat it that way, and then you just do comparisons to that, exactly. So that's like the baseline. If you're the same as that, then you're true positive. If you're different, then you're either false positive or false negative. So that's sort of that's the validation approach, yeah. And I mean, I think it's been nice because it's an iterative approach. So like they've had a ton of releases of the genome model validation sets, and like every time people compare it against their callers, and they find places where it seems like my caller is correct here and you're not correct, and then it's something they can revise and improve. So it's been good, sort of really open community trying to make a unbiased data set that actually represents what's happening. Cool. Are there any questions from online? Is it event okay? Can people hear and all that stuff? I've seen people talking on on chat, so that's good. Thank you. Great. You can talk out loud too if you want. But yeah, I hope this was cool. I mean, I'm really happy people came to hear online. I think we'll try to see how we can figure this out for the future talks as well. And we'll try to see what we can do. If people online are interested in talking at this, we would also thought it would be cool to have a remote speaker. So that would be cool. We could change. I'm sure that someone might bump their date to speaking later if someone wanted to. We have an online schedule of all the plans and stuff. So as much as we organize ourselves, that's kind of how we do it. But and it's been okay for people here. It's not that different. It's not that weird. Thank you. Cool. Thanks, guys. Thank you. Works. Oh, I gotta turn off. Forward.